question to the audience. Can you hear any sound? Could be music playing.
Animesh, Erica, Shaden, Lisa.
Hello.
You guys
Get
can hear me
started.
okay, right?
Yeah, I can hear you.
Okay.
Get started soon. Waiting for people to trickle in. We had a lot of people. We had 8 students respond. So yeah, we'll get started. That's like on top of our normal uCHARM people. Hi, everyone. We'll get started at 5:05. We have some time for people to trickle in because classes must just have ended. So, hello, everyone. Welcome to our uCHARM event. This is actually our second event. The first one was with Dr. Laura Khan from Princeton University. But this is a really special event because it is done entirely in conjunction with uCHARM, which is sort of our parent organization, right? And yeah, I'm just going to quickly tell everyone what uCHARM is, and then we can get started with the really cool talk we have today with Dr. Victor Nizet. So uCHARM, it's an undergraduate club at UC San Diego. We have two principal goals. The first goal is that we need to educate the community that the threat of drug resistance is indeed a very serious issue. And the second goal is that we, like the generation that's in college right now, we can really do a lot of things to prevent the fallout from the spread of drug-resistant microbes. To that end, we want to encourage people to go into careers related to drug resistance. And we also want to equip people with the necessary skills to do that. And so as a part of that, we're hosting many webinars. And today's webinar is by Dr. Victor Nizet, who's a distinguished professor of pediatrics, pharmaceutics, and pharmaceutical science at UCSD. And he is the director of uCHARM. And he was one of the first people that I met when I was working on starting this club. And yeah, I think this will be the first formal talk about uCHARM that undergraduates will get to see. But beyond that, his talk will also feature a lot of things about how the current pandemic is very likely to worsen the situation of drug resistance. So yeah, let's give him our full attention. And thank you for speaking today, Dr. Nizet.
Okay, well, great to be here, Animesh, and all the other people who are members and interested in uCHARM. We're really excited to have this companion undergraduate organization. Of course, everyone's been thrown for a little bit of a logistic loop with the COVID pandemic, but I think it is having an effect to increase interest and understanding in infectious diseases, the need for therapies, how they're not immediately at hand, the challenges put forth by epidemiological considerations, global differences, educational differences, how hope can appear and then be peddled by some for financial or political advantage, and then scientists and physicians who many of career paths that many of you are considering need to get out there and be knowledgeable and vocal and put a human face on these issues for us to be successful, demystify things in an era of kind of increasing anti-science forces. So what I thought with my goals, and I'll share my screen, was to give an introduction to the uCHARM initiative. And I think on the call we also have Erica Anderson, who is a PhD scientist with broad experience in antibiotics, host pathogen interactions, and genetics and high throughput technologies, both in academia and industry. And so she's going to be a great resource for everyone, and has been putting together the events that we have for uCHARM. But I give you an idea of what inspires us. There's 80 different faculty who are part of uCHARM. I won't be able to cover all of them, but I'll talk a little bit about some of the research that's going on around campus, some of the things we have in mind doing educationally and programmatically. And then, as Animesh alluded, I'll wrap around to the COVID pandemic and how there are some, vicious cycles in which antibiotic resistance is making treatment of COVID patients harder, and the COVID pandemic is likely aggravating the antibiotic resistance crisis. And then, happy to answer questions, either along the way, if there's a mechanism of halting me, just let me know, or, at the end, certainly. Okay, so let me expand the screen, and I need confirmation from you that everybody can see this.
We can see it.
Just fine. Okay, good. All right, so uCHARM is an acronym for the Collaborative to Halt Antibiotic Resistant Microbes, and right there in its name, the point is collaborative. That's one of the special things about UCSD, in that there is a fantastic campus where the health science campus is immediately adjacent to the broader university with outstanding engineering school, basic sciences in biology, chemistry, the Institute of Oceanography. And so the medical school and the pharmacy school and the hospitals can closely interact, and many of us do, across campus in broad collaborations, which is really the only way to do anything that is truly revolutionary, certainly to translate new discoveries into therapy. And we debuted, just in the fall. So let's see, I need to use this, right? So our mission statement is we want to leverage and deploy the unique multidisciplinary environment, resources, and scientific talent of UCSD to lead innovative, and we can't emphasize innovative enough. UCSD as a university has a motto of we break things better, breaking the mold and going in new directions. But these should be research driven and ideally shift the entire paradigm about how we approach and treat infectious diseases to address the global crisis of both antibiotic resistance and the continual emergence of infectious diseases like COVID-19. So, antibiotics are one of the greatest success stories of modern medicine, maybe only second to sanitation, as having an impact on public health. And really, antibiotics have cured more patients than all the other drugs combined. And there was a dramatic reduction in deaths due to infectious associated conditions, which were common causes of death before, even in advanced countries like America, where people would die of scarlet fever and diphtheria, and life expectancy wasn't all that high. But antibiotics came and allowed a treatment approach to kill organisms. But in the last number of years, this has become increasingly difficult due to basically Darwinian evolution of the pathogens to acquire resistance to the drugs. And a factor that is compounding this crisis is that the drug companies who in the 50s and 60s were discovering a large number of antibiotics really have left the field largely. And the main reason that they leave the field is because they don't view this field as particularly profitable. Because compared to other drug classes like a blood pressure medicine or a diabetes medicine or a cancer medicine where you give very high prices you can command for the therapies, patients are only treated for a few weeks with the antibiotic. And then the very issue of resistance leads to a disincentive for the companies, because if you spend what takes about $3 billion now to make a new drug through all the clinical trials, and the organism evolves resistance to it in a few years, then it's very hard for you to recoup that investment. There are many causes of antibiotic resistance. One huge problem is overprescribing of antibiotics. This is simply a lot of people are given antibiotics for non-bacterial infections for which there's no specific antibiotic treatment, like the common cold, infections that would go away on their own. That exposes the bacteria in the body to the antibiotics, selecting for resistance, and that can spread in the community. And then, unfortunately, another patient, maybe one whose immune system is lowered or who had surgery, can have a serious consequence of that. Maybe about a third of cases overprescribed. And patients aren't that compliant with antibiotics. If you kind of start and stop antibiotics or underdose antibiotics, that can allow resistant bacteria to kind of train themselves and become more resistant. Many people are not aware, but you should be, that even more antibiotics are administered in the agricultural industry than in human medicine, especially in raising livestock and fish farming. The antibiotics are used because they promote growth of the organisms. They modify the microbiome and promote growth of the livestock and the fish, so they reach market size sooner. And then, unfortunately, those antibiotics are seen by bacteria in the environment and in the foods themselves that can then make their way into people and into human medical problems. Infection control sometimes falls short in hospitals. and infections get spread from individual to individual. This also occurs a lot in chronic care settings and even daycares. And in the developing world, lack of hygiene and poor sanitation just makes this a urgent problem. So even though we might start the antibiotic resistance in countries like America, where we do high-level ICU care and expose them to a lot of antibiotics, those can, in a world with global travel, that can spread to developing countries. And in the developing countries, they may have fewer antibiotics to treat, but it can get into the water supplies and contaminated. And in places like India now, you have areas where people are coming in, they've never been to the hospital, and they have a multi-drug resistant pathogen that is resistant to antibiotics that have never even been used in India. And so you can see how that fuels things. And I mentioned already the drug companies have gotten out of the field. I'll also talk about how drug companies haven't been very creative in antibiotics. They've basically just made me too chemicals that are similar to the pre-existing ones. And of course, when you just keep rewriting the same playbook, resistance is easier to develop among the pathogens. So fortunately, a number of governmental organizations and public health organizations have been alerted to and are trying to mobilize around this issue. You know, the most serious form of infection, which is called sepsis, is now estimated to be responsible for 20% of human mortality. That exceeds cancer. There's not a single approved medicine for sepsis beyond antibiotics. There are 0 novel antibiotics currently in the development that have the potential to treat the most dangerous forms of drug-resistant bacteria that are out there. And COVID-19, you know, the example of how a new infectious agent, which could be bacterial or viral, can pop on the scene. People don't have prior human immunity. There's no antibiotic or vaccine immediately available. And you can see how that can cause a lot of spread and economic harm while we are playing catch up. There was one study commissioned by the British government that estimated that on our current trajectory, that by 2050, antibiotic resistant death will be, you know, on a level exceeding cancer, and maybe at a cost of 100 trillion in lost economic productivity. The United Nations convened a panel of, or a General Assembly meeting, only the second time General Assembly has been dedicated to an infectious disease issue. Hopefully COVID will be the third. HIV was the first. And all the member nations signed on to say that this is an urgent problem. The Obama administration created a national action plan, which was updated prior to his departure. There hasn't been much further work on that recently, and the World Health Organization is very concerned and sponsoring a lot of global mobilization. So here's an example of the kind of disease that we see that's so scary. Many of you have heard about MRSA. This is Staphylococcus aureus, the most common cause of infections in hospitals and communities in America, really, of serious infections. It causes skin infections, it causes bone and joint infections, it's a leading cause of bloodstream infections. It can cause an abscess or a pocket of infection anywhere in your body. And resistant strains now account for about 50% of the isolates that people see in the hospitals, which makes treatment that much harder. This image here is of a child that we took care of back in the early 2000s. Bryce, who was hanging out with Santa in December, but then in January found himself in the ICU at our children's hospital, clinging to life with a community-acquired methicillin-resistant Staph aureus pneumonia. And I can show you his picture and give his name because he and his family have been advocates for the Infectious Disease Society of America awareness campaign, Bad Bugs, No Drugs. Tuberculosis is a pathogen that infects actually a third of the world's population, maybe 2.5 billion people have tuberculosis, but it usually lays latent in the body, but it can reactivate at any point during your life, especially if you have other immune problems, like there's a lot of synergy with the HIV epidemic, and causes pulmonary disease, but can also spread to other parts of the body. And this is an organism that is very slow growing, requires multiple antibiotics to treat, and we've seen a lot of extreme drug-resistant strains for which successful clearance is almost impossible. The ICUs and the chronic hospitalized environment, patients who have to come in often to the hospital because their immune system is lower or they need multiple surgical interventions. We see bugs like multi-drug resistant Pseudomonas. This is also a cause of chronic infections in people with the most common genetic disorder in America, which is the cystic fibrosis in the lungs. It also causes burn infections, and these strains are highly drug resistant. Sometimes in the CF patients become resistant to all antibiotics. Another group of organisms is carbapenem-resistant Enterobacteriaceae, like Klebsiella and Enterobacter. Carbapenems are a last class of broad-spectrum antibiotic that we were really reliant on, and now these organisms have developed ways to break down those carbapenems, and we had an outbreak in our own UC system at UCLA with half a dozen deaths associated with these organisms spreading from patient to patient on medical equipment that was being used to look like colonoscopy and duodenoscopy to look inside the patient for gastrointestinal diseases. They were acquiring the organism and then developing infections that were impossible to treat. A bacteria that's not really antibiotic resistant, but kind of bears mention in this area is Clostridium difficile, or familiarly called C. diff. This is an organism that all of us have a couple of these guys, hanging out in our guts at all time. In a normal, healthy person with a normal microbiome, that's not much of a problem. But when you start to take broad spectrum antibiotics, you can knock out the flora, and then this bacteria overgrows, and it produces a very dangerous toxin that can basically eat away the lining of your gut and produce something called toxic megacolon, which just by its name, is not good to have. It's actually significant mortality and a big problem in hospital. And then STDs are becoming extremely drug resistant, in particular the gonococcus that causes gonorrhea, or the clap. This is a sexually transmitted disease that is now very, very difficult to treat. Men are usually symptomatic with painful burning urination and discharge, but women sometimes can have the disease, and it smolders up in their upper genitourinary tract without coming to their attention, and some of the consequences might be chronic inflammation of the fallopian tubes, which leads to infertility. So with those kind of bugs, you know, we recognized that we had a lot of strength here, that this was going to be a big issue for human medicine. So we started sampling interest around campus and discovered about 80 different faculty who are doing research that touches on the antibiotic resistance crisis from one way or another. And by bringing them together, I think we could make a very compelling case for UCSD being an international leader in the field. And both the Vice Chancellor of Health Sciences, the Dean of the School of Medicine, and the Chancellor of the whole university, Pradeep Khosla, have been enthusiastic and given us some seed funding to see what we can do with community building events and networking like this. And of course, our major immediate achievement was the recruitment of Erica as the managing director. Well, you are all UCSD students, I imagine, and know that we have an amazing environment right at the very top nationally in terms of research funding and very influential scientists. So here is the place to do it. And the members of uCHARM are not just in the School of Medicine, like most people would assume, but in the pharmacy school, in the School of Engineering, in biology and chemistry, and in the Institution of Oceanography. We have multiple faculty in all of these elements of the university, so it's truly interdisciplinary. And Erica, you see there, is the managing director, and then we have a number of other key people who are involved in future fundraising, developing educational initiatives, getting the word out when our scientists have cool discoveries, and coordinating our events and grants. We have kind of a broader leadership council among the 80 faculty. These 15 faculty work with us on the high-level strategy of the center. And I think, you know, each one of their inputs is very valuable because they know about UCSD in different ways. They're all collaborative scientists. They have different areas and perspectives of expertise, and it's great to bring them all together. So what we really want to emphasize about uCHARM is, you know, there's obviously other antibiotic resistance efforts around which, you know, are involved maybe in the epidemiology or documenting the economics of the crisis, or doing clinical trials of, you know, the next new antibiotic. We thought that what we're really good at UCSD, and honestly what excites the faculty that are participating in this, is to really try to look at this problem and say, you know, we're kind of dug ourselves a hole where we only have a, you know, one kind of group of drugs, chemicals that kill bacteria in a test tube, the bacteria becoming more resistant. It's obvious that we have a lot of treatment failures, that innovation has been low in the field, mainly because of the departure of a lot of the pharmaceutical industry. And maybe we could take advantage of all the brilliant minds here and come up with real new ideas on how to deal with the crisis. So some of the ideas include, if you figure out how the bacteria produces disease, those are called the virulence factors and virulence mechanisms. Instead of just trying to kill the bacteria, which has a negative effect on all your normal bacteria too, we could strip the bacteria of their virulence as a treatment. Then the bacteria become harmless and your immune system has no problem with them. Also, we're exploring a lot of drugs that would boost the immune system function to increase the ability of your white blood cells or your epithelial barriers to produce antimicrobial factors that defend against infection. Our university has outstanding computational biologists, and some of our biggest investments in this area have been in applying big data, multi-omics, and systems biology analysis to the question, giving entirely new perspectives, especially when those scientists work closely with the doctors and biologists. We have an amazing center of microbiome science and innovation here, and we want to harness the microbiome through strategies of probiotics and prebiotics to preserve its integrity to fight off infection. Also, the microbiome is a very good source of new chemical diversity that can potentially be used for targeting bacteria or boosting immunity. We're right on the ocean. We have the nation's leading oceanographic research institute. 70% of the chemical diversity in the world is found in the ocean, and it's largely untapped, so we can find a lot of new drug candidates there. Chemical genomics is a way of combining high throughput screening with whole genome sequencing to understand the bacterial's capacity for resistance and be able to thwart it in advance. We have engineers who are developing new nanotherapeutics or developing rapid ways of diagnosing bacteria without having to culture them. We have a phage therapy center that's basically taking natural viruses that infect bacteria and using them as therapeutics. And we have an amazing case of our own faculty member being saved from an untreatable infection and a coma that is now a major book and a movie under development that was actually the topic of our first uCHARM colloquium. Computers can model potential targets and screen virtual libraries of drugs. There are a lot of innovations in chemistry now that can be deployed to improve absorbability or reduce toxicity of drugs. Vaccines are obviously an effective way to prevent infection in the first place. So we need to maybe look at exploring new vaccines to reduce, to target superbugs or treat common infections to eliminate antibiotic exposure. CRISPR is all the rage, and we found ways of using CRISPR to scrub antibiotic resistance from microbes. Drug repurposing is a huge and maybe our only immediate solution to the COVID crisis, and we see how that has been beneficial with remdesivir potentially being repurposed from use as a Ebola medicine, and maybe the use of some immune modulating therapies targeting IL-6 or intravenous gamma globulin to reduce the inflammation in the lung in COVID. And all of these things need us to redefine the definition of antibiotic, because right now the definition of antibiotic is just a drug that kills bacteria in a test tube on bacterial agar or seaweed. But you're not going to discover something that neutralizes virulence or boosts immunity in a test that doesn't involve any immune cells and requires killing of the organism. So we need a new testing paradigm. So who are some of these scientists? Down at the ocean, we have the marine microbiologists and natural product chemists like Paul Jensen and Bill Fennical. They go explore. It seems like they're always exploring places like Fiji and the Bahamas. I'm not sure why they're not there off the coast of Newfoundland or anything, but they do end up discovering new microbes. In fact, they've discovered more than 20 new genera of actinomyces, which are famous for producing antibiotics, and new chemical diversity that we're screening for antibiotic and immune boosting activities. So here you see the kind of setup they have. They're able to culture bacteria that a lot of people couldn't culture from the ocean by using special conditions involving seawater agar and alterations in the pressure and gas composition. They do whole genomic analysis of the organisms and also stimulate the bacteria to induce their activity of these novel chemicals. Bill Gerwick and Brad Moore are two other marine natural product researchers who have unique approaches. Bill studies cyanobacteria, which are very interesting chemically diverse organisms that are often commensals of larger marine flora. And Brad Moore also has the research where he is using novel genetic techniques to unlock the biochemical potential of hidden natural product loci. Here's an antibiotic that Bill Gerwick found that was the largest antibiotic known. He called it gargantuolide A. From a novel streptomycin with 105 different carbon atoms. It seems to be really good against MRSA and Clostridium difficile. And here you see some work that we helped out a little bit with Brad Moore, where he found some genes in the sequence of the organism that looked like they should make a natural product. The bacteria didn't make it themselves, probably because we didn't have the ability to fully reproduce the conditions of the deep ocean. But when he was able to clone it out and optimize all the promoters and express it in either yeast or bacteria, and then you get the product produced, it turned out to be a very good antibiotic. And also, when you're making these genetic operons from these different organisms that make these complex natural products, you can switch and swap the different genes and maybe get new chemical diversity that never existed in nature, but might have a good chance of having a bioactivity that you want. In biological sciences, the Dean of Biology, Kit Pogliano, and her husband, Joe Pogliano, they are bacterial cell biologists who've developed this very novel technique called bacterial cytological profiling, where using fluorescent imaging and very high resolution and high throughput, they can quickly determine the mechanism of action of potential antibiotics, and this allows people to pick the highest yield targets, screen natural product libraries for activity, understand how unknown molecules work, and they've gotten it to work with a lot of the highest priority pathogens. Rob Knight and many others who've come more recently, like Jack Gilbert, are part of the new Center for Microbiome Innovation. David Pride is a physician scientist who works on the microbiome and also the viral communities that contribute to antibiotic resistance. And the microbiome is an unfortunate victim of overzealous antibiotic use. We can knock out the microbiome by giving too many or unnecessary antibiotics and set up a host of other health problems. For example, there's very good evidence that the map of obesity in America correlates very closely with the map of antibiotic prescriptions, because you screw up your metabolism when you screw up your microbiome. So we want smarter therapies. So figure out how you can study the microbiome in parallel with the development of some of these new therapies. Ideally, virulence factor inhibitors and immune boosters do not alter your microbiome. And here you can see this concept that, as infectious diseases went down with antibiotics, a lot of these other diseases like inflammatory diseases and allergy and diabetes have gone up. And there's a very prominent theory that it's all the missing microbes we've done by lining our gastrointestinal tract with too many antibiotics. They have very, very novel techniques where they can map not only the microbes, but the molecular composition in the body in 3 dimensions. And here you can see in cystic fibrosis patient who is getting a lung transplant that we were able to determine in the diseased lung exactly where antibiotics get in, where they don't get in, where the microbes are, how that correlates to antibiotic concentration, and local immune response. Sort of like microclimate weather that a local news station would advertise. That I think we need to remember that a patient is not just one vessel, but there's a lot of variation in the body that we need to take into account. Here you see some of David Pride's research, which is finding that that microbial diversity, including the diversity of the viruses and bacteriophage in your body, changes after typical antibiotic courses, and then can be spread from family member to family member. The chemists are really critical to this process. They use, a variety of classical and innovative techniques, including developing molecular probes and elucidating microbial biochemical pathways to target them with chemicals. So we're talking about new antibiotic targets that no one has discovered, or ways to get antibiotics to penetrate into the cells better, or to expand their target range, or to reduce their toxicity. Here you see some work from Mike Burkhart's lab, where they discovered that an enzyme was could be targeted and hijack that biochemical pathway to inhibit critical metabolism of Staph aureus as a novel therapeutic. And here's some work from Yitzhak Tor's lab, where he is modifying amikacin and tobramycin derivatives, some old antibiotics that have been tried and true, but resistance has evolved. And now these are able to once again kill the resistant bacteria. In the pharmacy school, we have more kind of medicinal chemists and chemists who are involved in high throughput drug design and formulation, like Jim McCarrow and D.O. Siegel. Jim McCarrow actually runs a center for discovery and innovation in parasitic diseases, neglected tropical diseases, like parasitic worms and that are in the developing world. And he has both the animal models and ways of culturing and studying the worms in the laboratory, because we believe our initiative is not restricted to the bacterial pathogens, but also includes viral pathogens, fungal pathogens, and parasites like malaria and these worms. Dio is a very ambitious scientist, and you can't show him a molecule that he doesn't think that he can synthesize from scratch, which is pretty impressive, because there's a lot of things that are discovered to have antibiotic activity, but we cannot get the organism to produce them in sufficient quantities, and that's exactly where chemists like bio break it down into bits, figure out how to put the building blocks together into an antibiotic molecule. And often along the way, you find that that a particular assembly or a particular intermediate is even better than the natural product that you started with. Another very important thing is using alternative models where we can do very high throughput analysis of drug candidates and learn about their biology and mechanism of action. So professors like Emily Trammell and Ethan Beer, they use worms and Drosophila fruit flies. Others use yeast as a model organism, or zebrafish. And just the fact that these organisms are, very small, you can do forward genetics. Discovery is accelerated, and they are very collaborative and work with the scientists who can extrapolate that into the mammalian models. Emily is working on a parasite that affects the gut, which is very, very similar to parasites that produce epidemic diarrheal disease in America and that are hard to treat with antibiotics. And Ethan, working together with our chancellor, helped inspire the founding of the Tata Institute. Many of you who know the biology campus know that new hall that was developed, and the active genetics technique, working with Ethan, we've recently got it into bacteria to scrub antibiotic resistance markers. Advanced technologies in mass spectrometry and the associated informatics in labs like Peter Dorstein's and David Gonzalez allow them to basically measure all the molecules in your body at once. You see Peter there mapping the molecular composition of the body in 3 dimensions. In David Gonzalez, he has a paper in that has great reviews in Cell, and where he was looking at the bloodstream of patients with Staph aureus to try to predict which ones are going to do well and poorly at a resolution that is probably 5 times what anybody had achieved clinically, and led to some predictions that we were then able to show in animals influenced outcome. Here's that lung study again, and some citations of many of Peter's ability to map the molecules of your body. There's a great article in the front of Nature, The Man Who Maps the Molecules, that you guys should read to learn about Peter's lab. Here is some work that Peter and Rob and Jack Gilbert are doing with the American gut to determine where are the antibiotics distributed in your body after you administer them. And shockingly, we're finding a lot of antibiotics in people who haven't even been prescribed antibiotics. So like 6% of people who never took antibiotics have these, presumably from dietary sources, or and that's worrisome that these antibiotics are in our food chain like that. Here's the kind of approach that David does to rapidly elucidate both the bacterial proteins and the host cell proteins throughout the body, and create a snapshot, even at an organ level, of how an infection is going. On the skin, we have Richard Gallo, the chairman of dermatology, and Carsten Zengler, who studies microbial communities. They are probing the natural molecular barriers, taking some of your healthy flora and using them to treat infection. And this is very important for staph infection and pseudomonal infections. they had a beautiful paper in Nature Medicine showing that commensal bacteria dampen skin inflammation, and that often infectious bacteria are trying to trigger inflammation to clear out the normal flora. So this leads to kind of a rethinking of what we need to do to prevent infections in the skin, rather than just sterilizing ourselves continually. We need to fortify the normal flora, which may be the most important barrier against infection. And here is some work from the Zengler lab, where he basically figures out how bacteria are participating with one another as communities. And only now, with this systems biology approach and the ability to do this kind of what you'd call metagenomics, even of bacteria that can't be cultured, he looks at the community of bacteria on the skin and their cumulative ecological potential, because the community can help one another out, where everybody is doing different parts of the metabolism. And if you study any one bacteria, you don't really understand the picture. And if you disrupt a critical element, you might lose not only that bacteria, but the whole community. Gural Soul and Partho Ghosh are biologists in the Department of Chemistry and Biology, respectively, but they have these fascinating biophysical approachs to studying host pathogen interactions. For example, Partho has elucidated the structure of key virulence factors of flesh-eating microbes that then lead to new vaccine targets. Gural Soul actually has a beautiful paper in one of the Cell journals this week talking about how bacterial biofilms communicate with one another through electrical signals that are very much like a neural circuit. And bacteria basically have a type of memory. Very provocative science. Sharon Reed and Lars Ekman, they are interested in doing drug screening for gastrointestinal pathogens, especially bacteria like E. coli, and parasites like Giardia and amoeba that you can get when you're traveling or on water exposure and are often drug resistant. And they found some, very exciting new drugs and repurposed drugs to treat these infections. Here's an example of new treatments for that were active against multiple different species of acquired GI pathogens, including Helicobacter, C. diff, and Giardia. We have Tariq Rana, who is the Vice Chair of Innovation in our Department of Pediatrics, and he studies pathogens like both bacteria, but also HIV and antiviruses using high throughput RNA biology and CRISPR techniques. Elizabeth Winsler does the chemical genomics and high throughput drug screening for malaria. Here you see that his discoveries in one of these screens led to a recognition that TLR3 activation is necessary for Zika virus infection of the central nervous system, and provided a way of inhibiting that activation, which might be good at preventing some of the neurologic devastation. Whereas Elizabeth has had high impact paper after high impact paper through the Bill and Melinda Gates Foundation Malaria Drug Accelerator that she studies, a number of which are entering phase 3 clinical trials, including the first drug that kills both the liver and blood stages of malaria. Don't want to forget the engineering school, including Animesh's research mentor, Liangfeng Zhang. We have his research on biomimetic nanoparticles, which can neutralize bacterial toxins. I have the very good news, Animesh, you might not have heard, but Liangfeng and I just got positive word on our what we call a CARB-X award, a grant worth up to $15 million to combat sepsis with nanosponges made from macrophages. Stephanie Fraley is a bioengineer who uses machine learning and nucleic acid-based technologies to do multi-pathogen detection on very small samples. Here you see like a drop of blood that you might get from a neonate, the ability by looking at different melt curves of the DNA amplified by PCR within a couple of hours, identifying what's there, and not only what's there, what its antibiotic resistance profile would be. And here's these nanosponges taking the membranes off of normal cells, coating biodegradable nanoparticles, introducing them in the body as decoys to absorb bacterial toxins and pro-inflammatory components. There I am, which means we're kind of wrapping up here, along with my colleagues like George Sakoulis, Monica Kumaraswamy, and others. We're in this new building on the medical school campus, and we're really working on trying to understand how antibiotics act in the context of innate immunity. Right now, the only testing we do for antibiotics is adding chemicals to the bacteria in agar, but that's not how the body is made. And we think that the fact that we don't consider the immune system and the chemical composition in the body in our antibiotic evaluation is a big problem, because before the patient has ever seen the doctor, their infection is already being treated by all these natural antibiotics, and we need to understand how they work, and we need to understand how our drugs might work in synergy with them. So we have an immune system. The immune system determines whether you get sick or not with your antibiotic-resistant infection, and yet their entire approach to antibiotic therapy now is ignorant of this biology. And we've begun to study that, study antibiotics, try to discover new chemicals that work at the host pathogen interface. Because we don't want to just define bacteria as sensitive or resistant based on the seaweed agar test. We want to define them as sensitive or resistant based on whether they work. The bacteria was sensitive if we gave the bacteria to a patient and they got better. The bacteria was resistant if we gave antibiotics to the patient and they got worse. And I think not doing this, we're shooting ourselves in the foot with the antibiotic discovery pipeline that we have now. So the old paradigm is to screen in the bacteriologic media. We keep discovering the same targets over and over again. But if we start understanding antibiotics in the context of human blood and the immune system, you can imagine we can discover all sorts of new drugs, like ones that inhibit virulence, ones that boost immunity, ones that sensitize the pathogen to be killed by immunity. We'll still discover the classical antibiotics, but why not, have your screens pick up these new ones too? And here's an example that we did research where the most common antibiotic prescribed in America is azithromycin, 60 million cases a year. But it's never used for these multi-drug resistant superbugs like the ones that killed those patients at UCLA who were getting the GI procedures. Why? Because if you do the test in the bacteriologic agar, azithromycin doesn't work. But our research showed that if you do the test in the context of the immune system helps the azithromycin get inside the cell and then it is able to kill the bacteria. And a single dose of azithromycin eliminated almost a 1,000-fold reduction of these pan-resistant bacteria in a mouse model and kept these mice alive. And this got some attention. There was some Iraq, stuff going on there, but we did have this research share the top cover, and then we've been doing a lot of work on superbugs and coming up with new therapies, and we want this kind of attention that we get locally because we're UCSD for the 80 faculty in our program to spread nationally. One thing that everybody's heard about now, and UCSD is the unchallenged world leader in, is phage therapy. This was our professor of psychiatry, Tom Patterson, who developed a life-threatening superbug infection with Acinetobacter, also known as Iraqi bacter because of its prevalence in the war theaters in Iraq and Afghanistan, and was medically airlifted back and was in a coma and is failing all antibiotic therapy. And in large part due to his wife, our Dean of Global Health Sciences, Stephanie Strathy's leadership, as well as that of Chip Schooley, then the head of infectious diseases. We received bacteriophage from all over the world, tried them, and got him cured. And they've been on, all the news shows, the BBC and the Today Show, and a book called The Perfect Predator is out that you guys should read. And there's a movie in the works. Apparently Will Smith's production company is after it. It's quite a story. So, as part of uCHARM, we have submitted what's known as a training program for physician scientists. This was the big haul that Eric and I worked on a lot. We're calling it CARING, Creating Antibiotic Resistance Innovators for the Next Generation. And this will support physicians and PhDs to do infectious disease research relevant to this. And based on that submitted application, we're hoping in the next year or two to write a parallel program targeting PhD students. Erica is also taking the lead in our developing an online master's course that is part of the new UCSD Strategic Academic Program Development. Maybe by the spring of next year, we hope to have one course and then develop it into 3 courses that can be integrated with master's degrees that people can get online here in drug development or personalized medicine. We had a big grant on the systems biology of infectious disease. We have another one due in a couple of weeks on a center grant involving multiple investigators. And I mentioned our big CARB-X awards that we got. This one we just learned is happening. So here's kind of the outline we had of our systems biology grant that was awarded a few years ago, and we're looking to continue that. This involves multiple labs all over campus. Another thing that we hope to do with uCHARM is develop lab capabilities that we can open up to the biotech community or to other labs that maybe have some interesting science that applies to antibiotic resistant, but they don't have the ability to do antibiotic models or test all the superbugs. And by setting up these core laboratories, our import in this fighting this problem can be expanded. All these scientists that I went through, we all interact with all the other existing research programs that have relevant topics. So we support one another across campus. And we have reached out and identified contact people at all the other private research institutes, Rady Children's Hospital, San Diego State University, and all these amazing international centers around the world, in Europe, Asia, Australia, and throughout America, that are doing something exciting in antibiotic resistance, that have agreed to be partners with us. And then the other key people, of course, is the biotech community. And here are a number of local companies that have been coming to our uCHARM events, and many of them have had featured speakers. These events, I've seen a number of you there. They're open to the general public. We don't know when they'll start again, honestly. All the speakers that were canceled in the spring due to COVID have agreed to come next year, but we don't know when, something like a scientific conference with 200 people in a room can happen. We might take them virtually, although there's, quite a few virtual things happening. We also have research in progress, meaning postdoctoral fellows and graduate student lunches. All these things have food, they have networking time, and undergrads are absolutely invited and encouraged. We're building a web page that will highlight all the different faculty researchers, all the different exciting approaches, all the superbugs. We're integrating that with some social media. Of course, we want to feature uCHARM there. There's also opportunities for people who are interested in learning a little and helping out with developing individual faculty web pages, etc. To volunteer, you can contact Erica. Okay, so I'm going to wrap up with just a little bit of thought about the COVID-19 pandemic. I'm not going to preempt my own infectious disease fellow, who is your next speaker in this series, who's going to tell you all about COVID. But all of us are at least mini experts on COVID from living in today's world. I just want to emphasize a few points about how the COVID pandemic is aggravating the antibiotic resistance problem. I think it's basically, kind of a vicious cycle. And you can see here that a lot of other people think that that's the case. We're seeing a lot of editorials and opinion pieces, both in the scientific literature and magazines and from agencies, about how COVID is likely to worsen the antibiotic resistance crisis, or vice versa, that antibiotic resistance complicates the management of the COVID patient. And one place that this starts is in recognizing that when you have pandemic respiratory infections, a lot of the mortality is actually driven by bacterial superinfection. And this is a paper by none other than America's doctor, Anthony Fauci, where he analyzed data from including postmortems, tissues that were available from the 1918-1919 flu pandemic. And these tissues indicated that a large majority of the cases actually had evidence of severe bacterial bronchopneumonia, with a lot of neutrophil infiltrates, and numerous bacteria were seen in the edema fluid and the macrophages. Of course, this was super devastating because this was pre-antibiotic era. And here was data in terms of identifying the culture of the organisms, where you could see in the patient's autopsy series that pneumococcus, Streptococcus hemolyticus, now known as group A streptococcus, and staph were in a large number of patients, and many of them had mixed infections with multiple pathogens. In terms of not having bacteria, very few of the patients died without having bacteria in their lungs. And this was borne out in the antibiotic era with the 2009 H1N1 flu pandemic. Now, that was initially considered to be a relatively mild epidemic, didn't really gain pandemic status, but CDC, did some analysis and felt that it was vast undercount, of the global mortality. I'm sure COVID is going to beat this, but they think that maybe up to 284,000 people died, and calculated that 50% or more of these infections were likely related to a bacterial pneumonia coming along afterwards. On data that is coming out so far in the COVID cases, it looks like we're seeing a significant amount of secondary infections, especially in the severely ill COVID patients in the ICU. 25% of them or so are having documented secondary bacterial infections. These are mostly bacterial or fungal. And the ones who end up on the intubated for a long time, they frequently develop multi-drug resistant gram-negative pathogens, including in China, a lot of them had carbapenem resistant Acinetobacter. And antibiotic use is very, very high in these patients because, and it's estimated that 70 to 80% of the patients that are in the ICU for COVID pneumonia are receiving broad spectrum antibiotics. And sometimes you just can't tell, from the x-ray whether there could be a bacterial infection. We know that when patients are on a ventilator for a long time, they're at very high risk of getting bacterial infections. And we're not able to do the normal tests that we might do to rule out bacterial infection, because that's often done by collecting deep sputum samples through something known as bronchoscopy. But, we're trying to protect the caregivers from getting COVID and COVID from spreading around the hospital, so we just have to give the antibiotics empirically. Also, at the other end of the spectrum, we have some ideas that are thrown out there, like that azithromycin could work for COVID. This is not true. There was the VA study that I think many of you have heard about, where there were actually increased deaths in the patients receiving hydroxychloroquine and no benefit with Zithromax. Just today, a paper with more than 800 patients came out in the New England Journal showing absolutely no benefit of these medications. Yet, when they're touted by physicians like Dr. Didier Raoult there, or politicians like our president, what's the harm? Give it a try. That leads to, questionable prescribing by physicians and a lot of potential for self-medication. Like in a lot of parts of the developing world, you don't need a prescription to buy antibiotics in pharmacy. And obviously that's going to drive antibiotic resistance when people are taking antibiotics for a misguided reason. Also, superbugs intensify the public health crisis. They tend to attack, in the same places that COVID is most dangerous, places where there are elderly people, places where there are a lot of people with medical problems in close proximity to one another, like chronic care settings, and places where there are immunodeficient people from the immunodeficiency of elderly or chronic diseases. So in these congested care settings, that's where we've already knew there was an antibiotic crisis. And now we see the COVID having its devastating toll. And old age and sepsis from bacteria or fungi have been tied to the poor COVID outcomes and deaths. So there was a study in the Lancet, reviewing data in Wuhan that found that basically among the patients who died, more than half of them had clear signs of sepsis and blood clotting issues. And, you know, this is being borne out in other studies. You know, the COVID can get you, but if the COVID doesn't get you and you've been on a ventilator for a long time, you're very, very high risk of these infections. So with those tie-ins and leaving 10 minutes or so for questions, I want to have some concluding thoughts. Basically, we have an antibiotic resistance crisis in part because physicians like myself and researchers and the pharmaceutical company and the public at large got complacent. We thought we had things covered with antibiotics, but the bacteria just kept evolving, and now we have multiple resistant strains and a big innovation crisis. And people are dying of antibiotic-resistant infections. And I want to contrast that with cancer, where cancer doctors and cancer researchers never had the opportunity to become complacent because there are so many cancers that have very difficult, you know, treatment opportunities are limited. The mortality is high, like pancreatic cancer or brain cancer. So the doctors, they don't just give up, they keep trying to innovate. And what have they come up with? Well, they've come up with a number of drugs like drugs that target the mutated kinases in specific cancer cells while leaving the normal kinases unaffected in your normal cells, like Gleevec that cured a lot of leukemias, or the immunotherapies. That's really the major advance in cancer in the last decades, and we have diseases like melanoma and certain types of lung cancer being effectively treated by either boosting white blood cell activity or inhibiting the ability of cancer cells to downregulate the immune system. Our former president, Jimmy Carter, had malignant melanoma to the brain at age 91, and he got an immunotherapy that cleared him 5 months later. If you were 19 years old 10 years ago and had the same diagnosis, you would have been dead 5 months later. So this is what we're thinking. You know, right now, part of the problem with the antibiotic resistance crisis is these are the only drugs we have on the right, antibiotics that just kill the bacteria. But if you think about the whole interaction and then you apply systems biology and high resolution proteomics and metabolomics and good old-fashioned hypothesis-driven research with both immunologists and microbiologists working together, look at all these other potential targets you could have, which would end up allowing the body to defeat the microbe. And these are the kind of innovations we envision ushering in a new era with a lot more hope. So I will stop there, stop sharing. And Animesh, I'll turn it over to you as the moderator, and happy to answer any questions on any topic. Or, you know, obviously Erica and I are also resources for undergrads who are looking to make a connection with a potential research mentor or get involved in education. And along the way, you can get a lot of firsthand perspective on careers as a scientist, careers as a physician, and meet people that are in all of our labs at the different stages, graduate students, postdoctoral fellows, medical fellows, pre-med, undergrads who are working as a, you know, recently graduates who are working as a technician. All of these people have the real-time information on, you know, how to do the next steps in your own careers. And it's great to network, obviously, beyond your peer group. And we look forward to providing those introductions.
All right, thank you so much for that, Dr. Nizet. And as he said, CHARM and its faculty can be a very good resource for undergraduates because the long list of names contains a very high concentration of illustrious scientists, physicians, engineers, whatever it may be. Like my PI, Dr. Zhang, he he's very well known in the nanoparticle world, and I actually found out about this after I started the whole uCHARM process. But there you go, he's a part of CHARM. So yeah, are there any questions?
Look at the chat.
Actually, Dr. Nizet, I had a quick question.
Yeah, please.
Yeah, what would be, in your opinion, as a physician, a good way to convince the general public that this is a pressing issue, both in the time of COVID and in general, without resorting to the sort of apocalyptic, like the apocalyptic narrative that we're all going to
Yeah,
die?
no, I agree. I think the apocalyptic narrative is, eventually it's like a boy, the boy who cried wolf type of scenario where, you see that with COVID, in our current COVID situation. Um, People are, doing the right thing, social distancing, following public health recommendations. And then the news on their TV is so dramatic over time that eventually it kind of deadens them, and you see this, sort of rebellion and counter narratives and pseudoscience arise. We risk the same thing with only providing apocalyptic scenarios. So I think you're right that physicians, it's kind of, I think, maybe a little bit more interesting for patients to, I mean, for people interested in the field, including the general public and undergrads, to kind of be invited in to think of the problem from the patient and doctor's perspective, because that's really what we're dealing with, a tricky problem. Here we have a person who is not doing well, who has some peril to their health, and we have limited options. And it's a little bit of a detective story, which I think a lot of people are interested in. It's a little bit of a puzzle solving, but it also involves communication and dialogue, continuously update on other options. And I think the the COVID pandemic is bringing home all the different elements that have to go together to solve a problem, and that people may have a better appreciation of the complexity of antibiotic resistance after this, and that there might be diverse solutions. But you're right, hectoring people and telling them they're bad. This includes also doctors, telling them they're doing things wrong when they don't really have any other options for patients in terms of overprescribing, and people are anxious about their own health and their children. I think you have to step back and appreciate that. So part of it is flipping, and a big part of our narrative is that we're focused on solutions, right? So the major theme of CHARM is looking forward to an era where there's more things that we can do about antibiotic resistance. And so I think we need to keep the communication message there. I think a lot of other movements, like Ryan White did for or Magic Johnson did for HIV, or Michael J. Fox have done for Parkinson's disease, is also putting, a personal face on this. So one thing that we've talked about potentially doing is creating public forums where we have individual patients that come, and we have a discussion with their audience where they tell their story. I thought it was very powerful, obviously, with Stephanie Strathy and her husband, Tom Patterson's story. But to do this with a variety of different, patients of different ages and different underlying medical conditions, and put them together. Here's a question. What are current restrictions for physicians in terms of prescribing antibiotics? It seems like from a patient standpoint, I can walk in with barely any symptoms and be prescribed antibiotics. You're right, there aren't really any restrictions to prescribing power of a physician for antibiotics in this country, and it's much more tightly regulated in other countries. Individual hospitals, like UCSD Hospital or Scripps or Sharp, will develop a formulary, meaning that they only have certain antibiotics that are in the hospital, and the committee will, avoid having, too many broad spectrum antibiotics. And they might flag certain prescription practices or high-risk patients, requiring you to get an infectious disease consultation in terms of your prescribing for that patient. And that is a field known as antibiotic stewardship, trying to control the prescribing practices in your hospital to limit the selection of resistant organisms and preserve the use of antibiotics for multiple patients. But in terms of, is there anything preventing me from writing any of you a prescription for any antibiotic right now? The answer is no, not by law. But in other countries, it's much, much harder. And places like Sweden and the Netherlands have much lower rates of antibiotic resistance as a consequence. Overprescription and poor infection control practices in hospitals and overuse in agriculture drive it. And we can make laws to curtail that if we want, but it requires, forward thinking. It doesn't involve disbanding task forces for public health and always bending over to the business interest, because the drug companies, they want you to prescribe the antibiotic as much as possible, and the beef and pork industry and chickens, they want to be able to do whatever they want to have their drugs go to the market. So if you keep turning over to the business interests, it's exactly like global warming. If you don't force the companies by laws to do things in the public good, you won't get anywhere. But America trails a lot of other countries.
Yeah, Dr. Nizet, there's two very similar questions, actually. Christine asks, just wondering, how do we get started in being involved? And Faith is asking, if they wanted to contact you, what would be the best way to reach you?
Well, I think the best way is first, through Erica. Erica and I meet regularly on all things CHARM. So the email address is charmhq for headquarters at ucsd.edu. Or obviously you can email me or Erica, and we'll circulate it to one another. And really what we want to do is make this personalized, just like we do with a graduate student who's contemplating a rotation or a new postdoc. We need to find something that is of your interest, that matches your available time in terms of schedule, whether you're coming at it from more medical perspective or a public health perspective or a basic research perspective, whether you're interested in biology, chemistry, engineering, pharmacy. And I think that a very good way is to get involved in research, in one of the laboratories. And the variety of research is unlimited. But then also volunteering in terms of the events that we'll develop, the development of the web page, and some of our upcoming public education activities could be in assisting us developing the materials for the course. These are all different ways that people can get involved, but we want it to be impactful for you. Of course, we want it to be personally rewarding, but we also, I think that people who give significant effort should be rewarded by having tangible accomplishments that they can showcase to help them in their future career development. So that's really the way that we want to do it. And of course, I think uCHARM has the ability to determine its own destiny. These webinars are a very cool start. We think that we can do a lot of things in terms of showcasing career pathways, but if uCHARM wanted to do something that was involved in community outreach that could leverage faculty participation in a way that is done in resonant with your mission, you're leading it, but we're supporting it, we're all game for that.
Yeah, and to kind of follow up on what Dr. Nizet just said, if you don't quite know what kind of lab you want to join or exactly what part of this whole, tackling this whole issue you want to be involved with, I would very strongly encourage you to join uCHARM, and we can help you figure that out, because we have a very diverse set of board. Like I'm an engineering major. I don't have anything to do with pharmaceuticals or biology except that I work in a nanomedicine lab, and we have students representing cellular biology, molecular biology, bioengineering, and pharmaceutical sciences. And we're also looking for people with a lot of different skills. And these skills, I imagine, like graphic design and stuff, they'll be transferable to CHARM. And there's a lot of interdisciplinary things going on. And just to start wrapping up this webinar, I would just like to thank everyone who came in, and please fill out this sort of exit survey so that we have a bit more idea about how we can improve these webinars. And lastly, I just want to say thank you to everyone, and especially to Dr. Nizet and Dr. Anderson, who took a lot of time out of their busy days to come in and give a talk. And here's the special opportunity that Dr. Nizet was talking about. CHARM is looking for volunteer authors to write profiles of their faculty for their website. And uCHARM can serve as a stepping stone towards that, where we can coordinate all of that, or you can directly contact Dr. Erica Anderson. And like Dr. Nizet was saying earlier, in this position, you would have a unique opportunity to interact with top researchers and learn about their science in a way that not many undergraduates get to. And if you're interested in this or want to learn more about it, you can feel free to contact Dr. Erica Anderson. I'll post your email in the chat, or you can contact me and I'll pass you on to the CHARM faculty.
All right. I look forward to meeting everyone in person when we get events going, or just 6 feet away from you in my office with our head coverings. So.
Yeah, thanks a lot, Dr. Nizet, for coming in to give us this very informational talk.
Okay, bye-bye.
Bye-bye.